Hi everyone, welcome to dot points P3, E1 and E2. So today we're looking at performing a first-hand investigation to determine the effect of light on plant growth. We're also designing and conducting a simple plant experiment in this case, and also outlining the role of control, randomization, replication, standardization of conditions in our trial. So first of all, we're going to have a look at the experimental design part of it. So when we're designing an experiment, what are the things we need to think about? So often when you get asked this question in the HSC, you'll have to design an experiment. You'll have to talk about all of these things. So as you'll see later, it's important that you do talk about each one of these things if you get asked a question about it in the HSC. So the first one is a control, and a control is something that's used to compare test groups against, and it's normally a recommended amount. So for example, if we are testing a new type of fertilizer, we might use the recommended amount on the packet of the one that we're already using, and we might compare that against the recommended amount of a new fertilizer that is being introduced. Or if we wanted to test what amount of fertilizer was best, then we might test the recommended amount as the control compared to a couple of different amounts, more or less, that kind of thing. That would be the control. It's something to compare against, and sometimes it's hard to work out what it is in an experiment, but most of the times it's pretty straightforward. So how it's addressed in this experiment we're doing, it's going to be full light. So kind of full light is, I guess, the norm for plants. They normally get full light, and the other two things we're going to test in this experiment is going to be half light or 50% light, and then we're going to test no light as well. So with randomization, what randomization is obviously choosing things at random. So where you place different trial groups, which garden bed you use for which trial group is chosen at random, and you can do that a few ways, but the best or quickest way might be just putting them into a hat and just pulling them out of a hat. And basically that helps reduce the bias of the researcher. So researcher might choose to favor a certain outcome and might accidentally or even purposely sometimes put or subconsciously put something that they think will work better in a better plot. But it's removing the bias of the researcher. That's what the point of randomization is. So how do we address it in this experiment? Which bed we're going to use for the light, half light and no light experiments will be chosen by pulling the location out of a hat. Replication is pretty easy, as it sounds like in the name. It's just repeating trial groups. So you shouldn't just, if you're testing amount of fertilizer, you're not going to try one plant with one amount of fertilizer, one plant with another amount of fertilizer. That would not be valid because you can get anomalies occurring, and one plant might happen to be just a better growing plant than the other one, and it might not get any fertilizer but might grow better. So by having replication, lots and lots and lots of replicates, it reduces the effect of anomalies, which is an important bit here. So the example of how we addressed it in this experiment is we had 4 rows of 20 plants in each garden bed, and that is good replication. You could also potentially have more. You could do the experiment multiple times. That would be another way of replicating. If you only had a small garden plot area to do this in, you could just repeat the experiment 2 or 3 or 4 times, and that would also suffice as replication. The last thing you have to always address in a scientific experiment is standardization. And standardization is controlled variables. So you have to be careful here that you don't get control up here and standardization and controlled variables confused with each other, because controlled variables are things that fit under standardization. Standardization is where you try and make everything standard or make it the same, and you do that by controlling the variables, and the control itself is a different thing up here. But the standardization is the thing that you need to think about when you're doing controlled variables. So for example, you're going to have the same soil, the same amount of water. you ensure that basically everything in the experiment is the same except the amount of light. That is the only thing that is different. Even the type of seeds are going to be the same. The spacing, all that kind of stuff is going to be the same. And the amount of light is the only thing that you change. So how do we address it in this experiment? You have the same soil, same water volume, same seeds used, etc. So that's basically E1 and E2 dot point, kind of done there. We're going to look at P3 in a bit more detail now, and how we did this plant experiment. So first of all, the aim of the plant experiment, which we are performing, is to determine the effect of light intensity on the growth of bean plants. So how much light plants get, and the effect that has on their growing. So we chose to use full light, no light, and then half light using shade cloth. And so I've just gone through the method here, and what I've done is this is kind of a way that you could answer a HSC question here. If the HSC question asks you, as one does shortly in the past questions, to kind of design an experiment, then this is how you could do it. By writing this out, and this shows how I've addressed standardization, randomization, replication, and control. So they've all been addressed here in this question. So we have, first of all, we had 3 garden beds that were chosen of equal size and equal amounts of type of soil. That was standardization. The plots were chosen for each light group randomly from a hat, so it was randomization. All plots were planted with 6 rows of beans, replication, with 15 centimeters between rows and 15 centimeters between plants as per recommended density, which is standardization. And then we varied the light in the following ways. Full light had no cover, 50% light had shade cloth placed on a frame, and no light had black plastic placed on a frame. And then the second last dot point 5, all beds were watered evenly throughout the trial, which is also standardization. And then lastly, full light was used as a control to measure against. So here's an example of a diagram of the layout of the trial. So we had 3 garden beds. You can see this is the top view up here, and the top view shows what it looks like from above. So we have full light, 50% light, and no light. This is without the cover so that you can see what's going on. And you've got 6 rows in each one that are evenly spaced 15 centimeters apart. And you also have 15 centimeters between plants as per the recommended density, and you have full light, 50% light, and no light. So it shows the standardization between the 3 gardens, because you have the same conditions for each one. The side view shows what it actually looks like. So full light, there's no cover, 50% light is covered with shade cloth, and then no light is covered with black plastic. And so obviously no light gets through. In terms of results, we have no light here, 50% light here, and full light here. So we haven't actually finished the trial yet, but this is what has happened in the past, and this is just a kind of written way of putting the results rather than measuring. So in the HSC, you're going to basically put this to words like this rather than putting specific numbers in. And I've also put some kind of analysis in here as well about what happened with each one and why. So the results show that initially plants with no light treatment grew the tallest. However, they were pale, i.e. not green, and elongated due to seeking but not finding light. So those plants that have no light are not photosynthesizing, so they don't have any chlorophyll, chloroplasts, and they're not green in color. They eventually died after finding no light and running out of seed energy. The next point is 50% light. So these plants grew fairly quickly, and they showed green color in the leaves and signs of photosynthesis. Eventually, some plants, but not all, reached maturity and produced flowers, and competition was strong for limited light, hence not all reaching physiological maturity and therefore not flowering. So basically 50% light, some of them flowered and some of them didn't. The last one here, full light. Plants in full light showed fast growth and deep green leaf color throughout the experiment. There was strong competition, but due to large amounts of light, virtually all plants reach physiological maturity and produce flowers for reproduction. So as you get more and more light, the plants are able to grow more and more. There's less competition between the plants for light because there's so much light available. And so that means that more that once you get down to kind of full light, more and more plants down here are actually getting more sun, which means they get to physiological maturity. So they are the results of our experiment. This one here, this past HSC question from 2016, basically they've given you the outline here of a trial that was conducted, and it says here, identify 4 features of the trial that contributed to the standardization of conditions. So it's just an identify, so you only need to identify or name those things. You don't need to do any more than that. So you can just go 1, 2, 3, 4, and how are things standardized, or how are variables controlled is what the question is asking. So firstly, 4 similar sheds were used with 5,000 chickens. That's standardization. Industry stocking rate was used for all of them. That's standardization. All chickens receive the same diet. That's also standardization. And 100 chickens from each shed were weighed, which is also standardization. The next question here, what is the component of experimental design? So we're talking standardization, randomization, replication, and control that involves management of variables, or the same as up here, control of variables, and the answer is standardization. Question 2014, question 3, which environmental factor would be most likely to cause seedlings growing in a greenhouse to appear pale and elongated? So the answer there is, if they're pale without green color and elongated, they're trying to find light, and they're not finding any because it's pale, no chloroplast, chlorophyll. And so the answer is that they have low light conditions. So the answer there is B. If we have a look at this one, which of the following graphs most accurately show the effect of light intensity on photosynthesis? So what happens with photosynthesis is you get to a point where it kind of maximizes, and then it drops off. Sorry, it plateaus. You won't find it dropping off because this is, remember, light intensity. And as you know, probably, I guess the more light you get, we tend to get heat as well. But this is just asking about light intensity and not asking about heat. So if the light intensity in the same temperature conditions occurs, it peaks to a point and then it levels off. So the answer is C. It's certainly not any of these other things. If it was saying light intensity and heat plus heat, then the heat would cause the photosynthesis to drop off because the plant starts to shut down because of light. But the answer here is... So this next question is explain the importance of using standardized conditions and a control when conducting agricultural trials. So there's both standardized here, and control is what we're looking at. We need to talk about both of those things. So a control is used as the basis for comparison in a trial. It is normally no treatment or is the recommended amount of something, example, a fertilizer. Standardization involves control or controlling, making the same all variables except the independent and dependent variables, and this ensures the results are solely due to the effect of the independent variable and nothing else. The next question, 2013-7, what is the role of standardization in experimental design? To reduce the effects of biological variability, that's not the case. To reduce the effects of bias by the experimenter, this is randomization. To provide a basis for comparison between experiments, that's not the case. And to ensure that factors other than the variable under investigation do not affect the results, that is the correct answer. It is D. The next one. So describe an experiment. Describe an experiment to test the effect of plant density or growth on yields. In your answer, show how the experiment demonstrates sound experimental design. So I've given the aim here. This is the density experiment, remember, not the light one. So the aim is to determine the effect of planting density on the growth of strain prime hard wheat. And I've given all 4 things: standardization, replication, randomization, and control here, and shown how I've done it, which is really important. You can't get your 6 marks here unless you have all 4 of these things. So I have replication, randomization, replication, randomization, standardization, and control. So we've got 1, 2, 3, and 4. So it's really important that I have all 4 of those things there. and you can read that as well. In a research paper, what is the principal reason for including a section that details the research methodology? So why do we talk about in a research paper how we do the experiment? Readers can interpret the results of the research. Well, no, that's not the methodology, that's results. Researcher can patent the research methodology. No, they can't do that. It allows the reader to evaluate the validity of the research methodology. That is correct, because it's to check if the researcher has done a good job of doing the experiment correctly in the terms of the standardization, randomization, replication, and control. And it allows the readers to make informed opinions about how the results were analyzed. And this is not about analysis, remember, it is about methodology or how the experiment was conducted. So it's definitely C. Design a trial to investigate the effect of light intensity on plant growth. Again, I've for 4 marks, I've done all the things here. So I said aim is to determine effect of light on plant growth, and then I've gone into detail about how I've replicated, standardized, randomized, and controlled in there. And the second last page, so they've given another example here of giving an experiment that was done and then having to analyze or evaluate the experimental design in this research. So it's worth 5 marks. So basically what they did is they had 2 different farms, so farm 1, farm 2, and they had only 5 trees, so 1, 2, 3, 4, 5. They only had 5 trees on each farm. And you'll notice here that one farm 1 was located in southern New South Wales with no irrigation, and farm 2 was located in northern New South Wales and it had irrigation. So what they're doing is determining the effect of irrigation. Now the problem is that these farms were in different locations, and so the standardization is very poor. They needed really to have the same farm, the same place, the same climate with irrigation and no irrigation. So that's the first thing. So you'll see down here that standardization was not done very well here because there was 2 different farms used. One had irrigation and one didn't. And then also I wrote that it's poor. So remember, this is an evaluate. So I used the word poor and I underlined that. And I also said that the replication was poor here because they only did 5 trees on each farm, which is not very good replication. You really need to do more. And so then I've kind of gone and evaluated it and said down here how you would fix it. This last page here, again, this is giving you an experiment that someone did and kind of getting you to critique it. And it says explain how an alternative experimental design for this experiment may improve the validity of the results. And remember, this always comes down to standardization, randomization, replication, and control. That's when they're talking about validity of results, experimental design. It's always these 4 things. So you need to make sure that you do these things when you're analyzing the validity of an experiment. So it says a researcher in the field conducted a field experiment to determine the effects of plant density. Treatment A had 20 centimeters between plants, treatment B 50 centimeters, and treatment C 80 centimeters. And so you can see up here we have A, all grouped together. Down here we have B grouped together and C grouped together. Now the problem with this experiment is it was done on a sloping land. So we have like top of the slope, mid slope, and bottom of the slope, so kind of a hill. And then we have different soil texture as well. So we have sandy loam, then clay loam, and then clay. Now that's really terrible scientific design. The standardization is, and as I wrote here, the standardization and controlling of variables is very poor. They have all of these things grouped together instead of spreading them roughly around the place to mix up the conditions. They put them all together. So all of the A ones are in sandy loam soil, all the B ones are in clay loam soil, and all the C ones are in clay, which is not obviously very good for standardization. The variables are not controlled. You're also testing not only the gradient, but the soil texture as well as the density up here, which is not good. Another way of improving, so first of all, they could have used a property that had consistent texture across the whole field and also a consistent slope rather than a variety of conditions. Another way of improving the field would be to use randomization by randomizing these. So you might have had A here and then B, C, C, A, B, or something like that. It might have been randomized here. and that would help alleviate this if this was the area that you had to use, rather than grouping all the treatments together. So that is one way of improving that as well.